Thank you. Hi, I'm Elizabeth Lipke. I'm a professor at Auburn University and also co-founder of VivoSphere, where we're looking to make synthetic tumors for high-throughput drug testing. So long a time ago, when I was a graduate student, I went to one of my colleagues who was working, I was working on tissue engineering. She was working on cancer drug testing and understanding mechanisms of cancer. And I asked her, can we dissociate the cells from my brother's tumor and decide which drug to test next? And she said, no. She said, She said, patient outcomes do not are not reflected in vitro drug testing. And so for the last 20 plus years, I've been highly driven to change that equation. And very fortunately for me, I've been joined in this journey with Dr. Yuan Tian, who is my graduate student and now co-founder of our company, who co-invented the VivoSphere technology, and who is working with me to bring this to the marketplace. When we think about the problem, it's not just 2D cell culture and patient and those two things not correlating with each other, but it's the other platforms along the way. We know that 3D cell culture in animals have better recapitulation of patient tumors, but they also have problems with reproducibility, and high throughput is not possible. They also then are still not completely reflecting patient disease. VivoSphere is working to bridge this gap in a simple but not simplistic manner, with the effort being towards more accurately predicting therapeutic response and combination drug therapy responses during the preclinical phase. When we look at the market, R&D returns for pharmaceutical companies have been going down, and they're looking specifically at this in vitro market to drive those returns higher. So right now it's a $200 million market, but the piece of that pie, overall pie of $250 billion, is only going to get larger with time. So what do we have in terms of our technology? We're combining biomaterial scaffolds that are tunable, regulatory signals, and human cells. This includes patient-derived cells and cell lines. And we're putting them together into VivoSpheres, 3D cell cultures that build on the technology that's been developed in the tissue engineering space, but doing it in a very high throughput manner. And we've accomplished this through our patented microfluidic cell encapsulation system. So the cells and the materials come into this platform, they're photo cross-linked and collected. This means that we can produce enough VivoSpheres in 20 minutes for the end user to go from the bench to their high throughput drug testing platform, or 1 per second, or 2 million in less than 3 minutes. 2 million cells encapsulated in less than 3 minutes. So this is very, very rapid. This is what you see when you're at the end of the process. Highly uniform tissues composed of cells and materials. We can combine different cell types. We can change the material properties to mimic the tumor microenvironment in terms of stiffness, extracellular matrix composition, and other factors that are present in the patient. And that enables us to more closely recapitulate human drug response, but still doing this in a way where we can then take our VivoSpheres and, as we've published, put them into high throughput drug screening systems for use in identifying therapeutics. So if you look at the competing technology, these are MCF7 breast cancer cells, and you can see them aggregating together, and they form these beautiful petal clusters. This is how breast cancer cells behave in the ducts of a breast, right? So this is normal epithelial cell behavior. However, when we switch to VivoSpheres, we see the cells are much more disorganized. They are looking more like mesenchymal cells. They look like the organization, structure, and honestly behave like cells in the tumor. And so this is where we're building. So we have control over that tumor microenvironment, which is all critical to recapitulating patient drug response. We can support multiple cell types, patient-derived cells, cell lines. We can encapsulate the cells and keep them in culture for up to 29 days or longer. So we can expand the cells in the system, or we can start at a very, very high cell density. In fact, we can encapsulate up to 60 million cells per millipolymer precursor. So tissue level cell densities very, very rapidly at high uniformity through our photo cross-linking system, making our platform very competitive for the end user. So why now? Pharmaceutical companies are coming together. They're forming microphysiological international consortiums to adopt these models into their drug testing platforms. The FDA has given this the green light. They want to see these platforms being used in the drug testing arena, both because other models don't work and because they bring things to market faster. And they're willing to accept this data. And we have a trend both in academia and in industry of reducing animal use. Why us? We have multiple cell types. Like I've said, we have numerous publications in this space, and we have 4 patents pending and 4 patents issued and 2 patents pending. So what are our revenue streams? Well, currently we're working on contract work to bring disease-specific VivoSpheres to the end user. This helps us work directly with them so that we see their needs and are building to meet them. This brings us initial revenue and credibility. Our next step is assay-ready VivoSpheres mailed directly to you through direct sales, through licensing. This brings scalable and continuous revenue, and we've shown we can do this. And then, for those people who don't want to let their precious cell lines leave their space, we're looking for instrument sales with consumables to build a long-term continuous revenue, coupled, of course, with databases that people can use to understand drug response. Why us? We have partnerships, like I said, with these end users, CFDRC, Southern Research, Thermo Fisher, in building the platform. We have academic collaborations, bringing people, publications and validation, basically showing that our product does what it says it does. And we've had inquiries, including, for example, from Crown Bioscience, who said that they're a contract research organization. They would like to buy the product if we could just send it to them. So what do we need? And this year alone, we've have over $500,000 in non-dilutive funding for the phase 1 SBIR and Innovate Alabama. And we have other SBIRs in the pipeline, but that is not certain, and the market is growing quickly. And so we're asking for $2 million to get us to this first quarter launch of our assay-ready VivoSphere kit in 2026. This will help us with R&D, manufacturing, IP, marketing, and team building so that we can bring this product to market with an exit strategy of acquisition by a contract research organization or pharmaceutical company with the goal of increasing their competitiveness and their throughput. And with that, thank you, and I'd be happy to take questions.
Couple of questions. Did you try using cells from PDX models?
Yes. In fact, we've published that data.
Does it work well?
It does work. And we're able to maintain both the stromal cell population and the cancer cell population. We have looked at multiple patient tumors, and when you do principal component analysis using transcriptomic data, the principal component analysis groups the tumors with the VivoSpheres, and we get good maintenance of the human cell population over time, so the cancer cells up to 29 days. And these VivoSpheres reflect, for example, the differing stiffnesses of the originating tumors.
Also, mammospheres, can you put them into animals and see the tumors coming up?
We have not tried that.
No? Okay. You think they would, right?
Yes, sir.
Okay, thank you.
Do your microspheres model angiogenesis?
We have previously encapsulated endothelial progenitor cells and supported endothelial progenitor cells in a large animal wound healing model in this system. And so if we were incorporating endothelial cells into these particular VivoSpheres, we probably could do that. We could also put them into other angiogenesis models, if you could imagine, for example, in microfluidic chip work that we've done in the past, research-wise, with CFDRC and their syn vivo chip. Thank you.
Anyone else? All right, thank you.